Hey, welcome to the Biochemistry Academy. In this video, we'll talk about triglycerol, which are formerly known as triglycerides. They are the esters of glycerol with fatty acid. The fats and oil that are widely distributed in both plants and animals are chemically triglycerol. They are insoluble in water and non-polar in character, and they are commonly known as the neutral fats. Now, triglycerols are the most abundant group of lipids that primarily function as a fuel reserve in animals. The fat reserve for normal humans in men is 20%, and that in women is 25% by weight, which is sufficient to meet the body's caloric requirements for up to 3 months. Storage of triglycerol. Triglycerol are stored in the adipocytes of adipose tissue, which are predominantly found in the subcutaneous layer and in the abdominal cavity. Structures of acylglycerol: monoacylglycerol, diacylglycerol, and triacylglycerol. In the monoacylglycerol, on the glycerol backbone, only one fatty acid is connected to the glycerol backbone. In the diacylglycerol, it is 2 fatty acids, and in the triglycerol, it is 3 fatty acids. Among these, triglycerols are the most important biochemically. Triglycerol can either be simple triglycerol or mixed triglycerol. In the simple triglycerol, they contain the same type of fatty acid, which are connected to the 3 carbons on the glycerol backbone, meaning that the whole 3 fatty acids can either be saturated fatty acids. An example of a simple triglycerol are the Triacylglycerol or the triglycerine. In the mixed triglycerol, which are more common, they contain 2 or different types of fatty acid residue. For example, the fatty acid which is attached to the carbon 1 is saturated, and the fatty acid attached to the carbon 2 is unsaturated, while on the carbon 3 can be either saturated or unsaturated. Stereospecific numbering of glycerol. If you check the structure of glycerol, it gives an impression that carbon 1 and 3 are identical. Although physically it might look like that, but that is not true in a 3-dimensional structure. In order to represent the carbon atoms of glycerol in an unambiguous manner, biochemists adopt a stereospecific numbering and prefix glycerol with SN. It should be noted that the carbon 1 and the carbon 3 are different. Cells possess enzymes that can distinguish these 2 carbons. For example, triglycerol is an example of such enzyme, which phosphorylates the SN3 and not the SN1. Properties of triglycerol. 1. Hydrolysis. Triglycerol undergo a stepwise enzymatic hydrolysis to finally liberate the free fatty acid and glycerol. The process of hydrolysis is catalyzed by an enzyme lipase, which is important for the digestion of fat in the gastrointestinal tract and fat mobilization from the adipose tissue. 2. Saponification. The hydrolysis of triglycerol by an alkali to produce glycerol and soap is known as saponification. Let's say a glycerol plus 3 molecules of sodium hydroxide will give rise to glycerol and soap. 3. Rancidity. The term rancidity is used to represent the deterioration of fat and oil, resulting in an unpleasant taste. Fats containing unsaturated fatty acid are more susceptible to rancidity. Now, rancidity occurs when fat and oil are exposed to air, moisture, light, bacteria, etc. In an hydrolytic rancidity, it occurs due to the partial hydrolysis of triglycerol by bacterial enzyme. In an oxidative rancidity, this is due to the oxidation of unsaturated fatty acid. This results in the formation of unpleasant products such as dicarboxylic acid, aldehyde, ketones, etc. It should be noted that rancid fat and oil are unsuitable for human consumption. 4. Lipid peroxidation in vivo. In living cells, lipids undergo oxidation to produce peroxides and free radicals which can damage the tissue. The free radicals are believed to cause inflammatory disease, aging, cancer, and atherosclerosis, etc. Tests used to check the purity of fat and oil. Because the adoration of fat and oil is increasing day by day. Therefore, there are several tests that are employed in the laboratory to check the purity of fats and oil. Some of them are 1. Iodine number. It is defined as the grams, which is the number of iodine absorbed by 100 grams of fat or oil. The iodine number is useful to know the relative unsaturation of fats, and it is directly proportional to the content of unsaturated fatty acid. Thus, the lower the iodine number, the less the degree of unsaturation. The iodine number of certain or common oils and fats are given below. Saponification number. It is defined as the number of potassium hydroxide that is required to hydrolyze 1 gram of fat or oil. Saponification number is the measure of the average molecular size of fatty acids present. The value is higher for fats containing short-chain fatty acid. The saponification number of a few fats and oil are given below.